I've been set a task, and I had no idea how I was going to introduce this talk until actually just over lunch talking with Michael Ediston. And he was talking about his boutique research that he does. So I think what I'm going to try to give you is a small snapshot into our collaborative boutique that we run here. So very briefly, the outline, as my talk says, our interest is from the host-pathogen interaction end. I'm going to talk about a study of just over 1,400 cases, and I've just split these into 3 study areas that we've been involved with over the last 5 years, 5 to 6 years. This is a neonatal sepsis, pneumonia, and measles vaccination. We have a very simple series of questions that we try to address in those studies, and I'll try to raise some of those challenges and the approach that we take for that. The first one is, what can we glean from just a single drop of blood in terms of information about what's going on in terms of a response in the infective condition? We're very much interested in actually how you actually do this type of science. So it's very much methodological based. We take a more systems level approach to this. Ultimately, the interest that we have is towards whether this has any translational value in terms of having more positive predictive value in the clinical sense from a diagnosis, but also in therapeutics. So just to put in the context, there's actually over 11 million deaths in children under the age of 5. This is attributed to a range of infective conditions. If this is broken down, you can see that neonatal infections account the bulk of this. This is seconded by pneumonia. So I'm just going to give you a snapshot into the work that we do with the neonatal unit in Edinburgh that we've been running for actually probably now 7 years. So the patients I'm talking about here are about the size of my hand. Obviously they're humans. They cannot tell you very much, they can't actually talk to the attendant neonatologist, the pediatrician, that they're not feeling particularly well. If you don't pick up the infection or infective signs early, you've lost a life within hours. Unfortunately, the current ways for picking this up in terms of support for the clinician to make a decision is about 15% sensitivity of the current diagnostic tests. So we were very much interested, again, coming back to not taking large blood volumes from these infants. They're currently a huge amounts are actually taken for current diagnostic purposes, so we wanted to scale this down considerably and to see whether, in fact, using the signals that we can detect from our own reactions and also learning how humans actually respond to infection in early life. So the test that we do is basically just on a little microchip, and so from a single drop of blood we will conduct on the order of 8 million experiments, and these are done on at a level that then you end up with a mass of data. Of course, this is unstructured information that we then structure it in the context of databases, and then put this, but of course, this is just a way of, if you like, getting information. But at that point, we also want to then bring in some knowledge, and so we use knowledge databases in the context of, and underlying what the division is about, is pathways, part of the biological processes for both disease and health. And so we use our pathway databases in an integrative fashion to interrogate this information. The point is that we want to move away from just a simple single signal detection system to really looking at how biological response. So just to give you a snapshot here, this is a sort of a case control study that we've done. Here we have the control, so these are uninfected, healthy infants and neonates. And over here we have the infected group in the red. What we were able to tease out from both using a variety of machine learning and other approaches for this was the coordinated action of a number of very specific and selective pathways. These are represented around innate, metabolic, and adaptive T-cell responses. The way that we approach this from being able to stratify the patients in that context is by combining these pathways, so we actually have an integrative approach in doing this. And when you do that, you actually have a positive predictive value that approaches 100% in terms of being able to diagnose the patients under those conditions. So that's just a little one snapshot of the work that we do. The other is here related to illustrating here in the cases with pneumonia. that account for a worldwide infection, like childhood deaths, of which pneumonia accounts for a large proportion of those. That's the green in here, and you can see that they top most of those as being the predominant cause of death. And so through collaborative work that we'd conducted with the MRC in the Gambia, in a study that we did here, you have a situation that a mother could potentially walk with her sick infant up to 3 days into the clinic, and the pediatrician has to make a choice as to whether to keep the patient in the hospital or send the patient back to the village, with the consequence that they've made a wrong decision and they've lost a life. So working very closely, we were interested in, again, whether we could look at the signals that you might detect at a systemic level in the patients for this. And so again, using this type of approach, we did this here for around about 400, well, just over 500 cases. And taking, if you like, those significant changes that we could see, and just asking now for all this morass of molecular data to organize itself, so to allow the data to sort itself out without giving it so in an unsupervised way. And this just shows a heat map of the actual gene activity that's happening. And so each one little speck here represents a gene in a particular patient that is shown up here on the top. What you can see is the color coding the patients here in terms of clinical phenotype. So very much interested in knowing whether this is a good way to use for phenotyping at a clinical level. And so what you can see is that the controls here are bunched along here. There's a whole bunch of genes that get down suppressed, and other genes here that are down that go up. What you can see is this gradation as you work through. The red guys here are the patients that are extremely, very severely, have very severe clinical signs of pneumonia. And so, and you can also see somewhat of a graduation response here. So we're greatly encouraged that this is going to be a very helpful approach for being able to predict whether you have a patient that will go on to severe pneumonia by staging them under this context. And so in that context, and just show you here is just looking at the principal components of variation that you find, again color-coding the control patients here, that they can clearly separate and stratify the patient groups with this. So the idea would be that you would have the mum walk in with the clinic, you're able to take a small pinprick of blood, be able to process that information quite rapidly and know whether you actually have a patient now that you spend more time looking after or not so concerned, and potentially make a decision with obviously other clinical signs to go back to their village. This is another snapshot of study that we've been involved with, and again, if you like, vaccination. A lot of vaccine approaches are based, the most effective vaccines are based on live attenuated pathogens. So these are, if you like, in themselves an effective event. It provides a very interesting window for research purposes as well in terms of understanding the response to an infective event. And so we've been conducting studies on the program again with the MRC Gambia on this, and looking at, and what I'll show you data on is some studies that we've done with measles vaccination, but this is the more studies for that. This is a study that we looked at in terms of just being able to, again, to look at the signals that you might see systemically in the time course. So this is 1 week, 2 weeks, 3 weeks, 4 weeks, 6 weeks, taken from infants that had prior to vaccination and then post-vaccination. And what we're interested in is what signals we can detect, and can these be predictors for response or quality of the vaccination response that's seen. I've just put here, this is a little bit more complex to deal with, and here we're also looking at, if you want, the time course of this response, of looking at those that are responders and non-responders versus the controls. And here you can see actually there's a lot of heterogeneity in the type of response. This heterogeneity we have to understand and be able to work with to be informative. What you can see is that most of the patients here, so these represent the individual patients. We're looking at a particular network or a pathway of signals that come up in those. And within the first week, you can see that most respond over here. The non-responders, you do not find this. And of course, you find a bunch of individuals sometimes that don't respond within the first week but respond in the second week and actually go through multiple waves. And so what we want to do over time is to be able to understand those types of responses. I want to end my talk, hopefully I'm on time, just to say that our approach here on this little boutique research end is that we use a non-living entity life form here for most of our basic research, as a viral particle, that actually has to plug and play with the host and the complexity of the host interactions that occur here. What we're very much interested in doing is picking up these signal changes that happen in those interactions, doing this in patients as well as in the test tube in our lab, using that information for both, if you like, exploiting new opportunities for diagnosis and coming up with innovative therapeutic approaches for the future. The premise for all of this is that actually we do a very good job at protecting ourselves against disease. And the idea is really, can we actually use those host interaction networks in that context? I would like to acknowledge my colleagues, in particular Paul Dickinson, who's co-leads with me on a number of these programs of activity in the division. Our collaborations with the lead consultant here, Ben Stenson, started off with an MD project in my lab with Claire Smith, and our MRC collaborations in the Gambia, in Fajara, Steve Howie, who's leading on the pneumonia, pneumococcal infectious work, Katie Flanagan on the vaccine work, and Richard Adegbola, who is now with the Gates Foundation. So with that, thank you very much for your attention.